So much for this invitation to be here. It's a pleasure to be here with all of you. And I will be talking about soils and microbes. And first of all, I wanted to acknowledge that the team of colleagues that I have worked with for the past 14 years that I have been here. I'm a soil microbiologist. I work at ARS. That's the picture of the building. It's across from UMC at 4th Street. And I'm adjunct faculty at biology and the plant and soil science department. And I work, I wouldn't be able to do this research without my technician, John Cotton. Terrence Garner is a postdoc that now is at North Carolina State University. And Jennifer Morkusera and John Sack from biology and plant and soil science here in Tech that I have worked with for the past more than 10 years. And Stephen Cox and some of the students that would be on their lab help a lot with this research, particularly that I will be discussing today on pyrosequencing. David Wester was at Tech and now is at Texas A&M in Kingsville. And generally the research that I have been working on is in terms of looking at the changes of the soil microbial component as indicators of soil quality due to management. We look at how management impacts soil microbes and how they can indicate changes in the soil quality and health. And generally over the years, I have established my lab in a way that I look at, when we look at the first component there, the characteristics of the microbial component in terms of the size, how many microbes we have, how the microbial community size is impacted by management. And we can do that looking at the microbial biomass, carbon and nitrogen. And then over the years, I also have been looking more at the composition. When we look at there, the fungal, the structure in this square here, we look at the structure in terms of fatty acid analysis. And I also have been able to get the lab set up and the analysis over time that we look at indicators, fatty acid markers for the fungal component and their fluctuations versus bacteria. We want to know if management is influencing, is increasing microbes, and also we want to know what type of microbes. So if we see an increase on the fungal component, we could most likely on the saprophytic, on the decomposers, we could have some indications that there could be more carbon sequestration, that we could be doing better in terms of nutrient cycling and increasing the organic matter content of that soil that could help other properties, increase the water holding capacity of the soil, the soil fertility. We also have some other indicators, other groups of fatty acids that can give us information of the arbuscular mycorrhiza fungal component that also helps plant to have a better plant water uptake and nutrients. And we also then look at their shifts and their changes with respect to bacterial populations. Over the years also, with the improvement on the DNA techniques and the high throughput sequencing approaches, like pyrosequencing is the first one that I have been using, but there are so many others now, like Illumina. I was able to also look more into the diversity, into what type of microbes are there to the species level. We say that we want to see increases on fungal populations with respect to bacteria, but all microbes are important. And we want to know then what are these fungal species that are being influenced by management and what type of bacteria. And what I want to know is how the microbial component is influenced and changed and impacted as they influence the metabolic capacity of the soil, the function. And I work more with the activities of enzymes, different reactions that we can track and have information of carbon cycling, nitrogen, phosphorus, and sulfur cycling. And as we look at the changes on the microbial component as indicator of soil quality due to the management here that since I moved, we had more cotton production and gradually we were seeing transitions to livestock, incorporation of livestock production, sorghum for biofuels, and have elucidated some of these changes. We have another layer of the climate also influencing microbes. And that's what I did some of the research on the record climatic stress conditions that we had on the 2011 drought. Many of you will remember on how we didn't even have any growing season, and the next year was also very difficult. By that time, we got record drought indexes And also the precipitation during the first 3 months by July 2011 were record low. Besides that, soil moisture content was very low for long periods of time. And this was really unique also because of temperatures 3 degrees greater than usual. So our soils for so long were being baked, and we really had up to 3 degrees greater temperatures than usual. So at that point, I thought we want to know how management is impacting microbes, and then we have situations like this in which we want to know what really happens there and how microbes then are impacted. If things were seen that are being increasing and beneficial for the soil quality, if this just goes down with these extreme conditions that we're seeing of climatic stress. So when we took soil samples, at that point, we went back when conditions improved, which was 8 months after, in March 2012. And that's when we saw some precipitation. We got some storms by that time, and we got some increases on the soil moisture that here really means a lot. And then also temperatures by then were better. So I wanted just to give a quick overview of the soils here, because we have then some of the soils more typical of this region that are generally a Pullman soil versus another type of soil that has just, here we can see an extreme of higher clay content, more organic matter, more microbes in that soil versus another soil that is then more sandy, higher in sand, lower in organic matter, and lower microbial communities. And we just tried to take soil samples at those conditions of the drought and heat wave versus when conditions improved on a rotation, and then versus a monoculture. And then that way, there is so little, there is no information available on how these climatic stress conditions really impact agroecosystems and how do we want to guarantee global food production, food security if we don't know these things. So what we found is that during that drought heat wave event, still the rotation will show greater levels of enzyme activities and the composition processes compared to a monoculture system, so management matters. There were still being fingerprints of better and more conservative management, and that would be then compared to the monoculture. However, what was then also interesting here is that with the high temperatures, record high temperatures that were reached, we will see the levels of the enzyme activities being so high. And then over the next samplings that we did over time, then they will be decreasing. But what this shows is that at that point, when we have climatic stress events like that, a lot of the organic matter is being depleted. And a lot of the things we're trying to do to conserve our soils could just be negatively impacted. So I just wanted to show that then we wanted to know then what were the microbes surviving these conditions in charge of these processes during that time. The first thing that we were able to see is that during a drought like that, we will see greater bacterial diversity indexes, and then they will just decrease by the next sampling time that we did when there were moist, cooler conditions. But then that fungi tends to show an opposite trend. They were then showing lower diversity indexes for the fungal component, and then they were just increased when conditions improved. So we just want to know on these different responses, what are the members of these communities that are there? And one overview that I wanted to give is that it is then possible that bacterial diversity indexes could be greater under these conditions, and the activities happening as actinobacteria were greater under the drought compared to when conditions were improved. We have here one soil type, the monoculture and the rotation, the other soil type here, and the monoculture and the rotation. So across these different soil types and the management, we could see increases on actinobacteria that are, many of their members are important in the composition processes. We also then found other members of bacterial phyla that is one that has been previously described as potentially resistant to extreme conditions, so they tend to increase during these extreme droughts in soils. Also, we found that for proteobacteria, some members within proteobacteria has been suggested to be also important in carbon and nitrogen cycling. And other group here also showed the same trend across soils and the management. Another thing that we found here interesting is that then they could be in charge of these decomposition processes generally under these conditions of stress, but what happens after? And there was just one group here of acidobacteria that has been suggested across the globe to be more predominant in soils that have low pH and be very critical for soil processes. But then in these soils here, we have alkaline soils. And we will always think, as we will be taking soil samples during summers, during the growing season, or just right after, we will always think that this type of bacteria will not be predominant in this type of soils, will not be maybe that important in soil processes. And it is because we never took samples over different periods of time. And then when we now did that, we will see how they will just be depressed, be decreased under these extreme conditions, but then they could really increase when conditions improved after. But what we need to think here on this dynamic here is how many of the substrates available are being decomposed, are being reduced over these extreme droughts and depleted in a soil. And then after that, then acidobacteria then could potentially recover and take care of processes after that. So it's just some of the implications of these fluctuations in the communities that we need to have here and understand, not only for these ecosystems, but for the nation, as we will experience more problems. I will skip this one, but I wanted to go into the fungal classes, and John Sack will help more on these implications, because even as a microbiologist, it's very difficult to understand the role of the fungal communities in a soil, and not much is known, and we also need to understand more of these, their response to this climatic stress that we're going to be seeing more with climate changes and the erratic the weather is. Like, I just wanted to point out that the diversity indexes will show that they were fungal communities lower under a drought like this and a heat wave, and generally it is then possible because this is just one group that were then greater under the drought and heat wave, and then decrease when conditions improve, but it only happened for one soil type. It didn't happen for the other soil type. Only this type of fungi group, droughtheat deomycetes, were then greater in the drought, and then they decreased when conditions improve, so maybe they're taking care of the composition processes of reactions when we have droughts and heat waves like this. We need to understand that better. And then, but this other group, more of them were then decreased under the drought and heat wave. And this is, for example, for the rotation for one soil. And then they increased when conditions improved. So this is also just a counterpart of what substrates are being used in a given time, and then when conditions improve, what other substrates could be available. The thing here is that we're trying to just look at what will be the predominant bacteria to the species level and fungal component also across soils and management during these extreme events, as we need to know that, we need to see if this means they're essential to soil function. I look for more collaborations to work more as these droughts could become more frequent across the nation. We also need to do more studies on their fluctuations in the soil for species that could represent pathogens and be pathogenic and have pathogenic potential. And also, as we were talking today about soil health, I also think about looking at the getting more information on the species that maybe are important for antibiotic sources, as we look at more of the problems that we're having now with the antibiotic resistance, that the president called for an initiative that could provide more information on that. So the climate will affect soils, and they're the source for microbes in the planet, and we need to have better information actually for agroecosystems. So any questions? Thank you.
down more thoroughly in your other papers in terms of separating the different aggregates and the aggregates within aggregates. And I just want to ask you, you didn't mention sulfur cycling here, and that the level of expression of sulfur, but it seems to be that what you're suggesting is that they're able to use the more refractory carbon component, that when moisture conditions improve, the fungi and the bacteria are able to use that carbon component. But why do you think that sulfur there's such an increased level? And then are you planning to look further into how these different aggregates are composed or disaggregate? What are the factors that cause microaggregates to form?
Yeah, it's more complex than as you are saying, that microbes are distributed at within different aggregate fractions. So it will matter more for soils with higher organic matter content than soils that are more sandier. And Jennifer has been doing studies on that, on doing more fractionation of the soils from this region and understanding the different, characterizing what are the different communities within different soil fractions. And this, for sure, then will matter also if you do better management, conserve more your soils through more conservative management, more rotations. Soils microbes produce enzymes that can become attached into the soil and be there for 10, 20, 50, 100 years and be a source of activities in that soil. And what we want to understand also is what is the link of the microbes to the production of these enzymes, to their different functions, because some enzymes then show different responses to these fluctuations. Like the one for the sulfur cycling responded differently because are more linked to the active microbial community in the soil. So that those are other things that we need to look at, the different processes and the changes of the different enzymes and their role in different nutrient cycling. That is more difficult to visualize on how the effect of climate will be. And when Michael was saying about the transport of microbes, that also is something that we will have to see how management would influence what travels in dust with the increases of the dust days that we're going to see also with climate change, that we also need to understand more. Any questions?
That's it. Thank you.
Thank you.